Good morning, IB Biology. This is Mr. McGee. I'm going to record this video for you on lipids. This is because of our unexpected asynchronous day that is coming with us here today. Keep in mind I'm from Michigan, so when we have snow days, it's usually a lot more snow than this, so this was kind of somewhat catching me off guard. I'm going to make this as quick as I can, and if you fall behind on anything, then you can always just rewatch the video, so that's why I'll try to talk faster than normal. What are lipids? Again, we talked about carbohydrates. Carbohydrates are sugars, and of course they have hydroxyl and hydrogen groups. Lipids, on the other hand, do not have such things. They're just hydrocarbons. They are generally hydrophobic, which means they repel water. They have nothing electrical that makes them stick to water. No charges, okay? However, they can be amphipathic in the case of a phospholipid. You remember that make up our cell membranes? Okay? They serve numerous functions. Of course, they store a lot more energy than a carbohydrate does, and that is, of course, because of all those CH bonds in them. Fat stores 9 calories for every gram that they contain, whereas carbohydrates are 4 calories for every gram. So fat is a tremendous source of energy storage. They compose our cell membrane and a phospholipid. Steroids are a type of lipid as well, so they can be used for hormone signaling. Waxes and things like that, like skin oils, can protect the body. Thermal insulation, body fat, of course, whale blubber, things like that. And of course, this goes without saying, but it also provides cushioning for the body. And let's be honest too, it also does provide appeal. After all, we do know that women tend to have curves. And that's not a coincidence. That's totally according to our DNA. So men are supposed to have more squared off features. Women have more rounder features. And that is, we owe that to lipids that does that. Okay? Whoops. All right. Energy in lipids versus carbohydrates. All right. Lipids are far more efficient at storing energy. We as animals, we use a lot of energy, so we need something better than glycogen. If we just used carbohydrates to store our energy like plants do, we would be much, much larger than we are now. We just wouldn't have enough space to hold it in our body. But fats allow us to have more of that density in us. 1 gram of fat, again, can store 9 calories of energy. Sugars, on the other hand, only 4 calories per gram. So you can see sugars, while they store energy, it's nothing like what fat can store. So therefore, we would rather use fat instead of sugar to store energy for our body. And let's look at a fatty acid. They all are structured like this. They all have what is called a carboxyl group at the end of it, which is also called an omega group. And if you recall, proteins have these as well, as does carbohydrates. This is basically a double bonded oxygen to a carbon, and then you have a hydroxyl group on one end here. We call this a carboxyl group, the entire thing. Now, in a fatty acid, it's referred to as the omega end. Some of you may have heard of fish oils before, like an omega-3 fatty acid or an omega-6 fatty acid, like in fish oils. You hear about these on commercials. Well, what that is it's, when we say it's an omega-6 fatty acid, it's 6 carbons down from the omega group. So it would be about this long. And if it's an omega-3 fatty acid, it would be about this long because it's 3 carbons down from the omega group. And that's all it really is referring to. We have a hydrocarbon chain, which is nothing more than hydrogens hanging off of the carbons. And although that seems really simple, remember, CH bonds store lots of energy, whereas carbohydrates have H's, but they also have OH bonds that don't store anywhere near the amount of energy. So fatty acids store these CH bonds. That's why they're so energy dense. Make sure you understand that. Okay, and they have that omega end. They also have what's called a methyl group on the end, but that's basically just where there's hydrogen all around this carbon, except for that other carbon-carbon bond right there. Okay, so these CH bonds store a lot of energy to them, and that is the key to this energy density. Make sure you understand that. Okay, and again, what's the difference between a carbohydrate and a lipid? So we have here glucose. And then pentoic acid, which is a type of fatty acid, which is, of course, fat. If we look at the differences here, they both look very similar for the most part. Now, you do remember that in sugars, when they're dissolved in water, they can actually form those hexagonal or pentagonal rings, or they can, when they're dehydrated, they can form these straight lines. So it really depends. For the most part, they look the same. However, there is a major difference here, and that is instead of having this omega group on the end of it, a sugar just has a double bonded oxygen off to one side like this. Okay? And the other difference is these carbons on the backbone of a sugar, they have these OH hydroxyl groups hanging off to the side, and they're alternating. Whereas in a lipid, it is just hydrogen and carbon. That's why we call these hydrocarbons. So please know that a lipid is also known, or fatty acid, I should say, which is lipids. They are known as hydrocarbons because they just contain hydrogen and carbon. And that is a very energy-dense material. Okay? They contain nearly twice the energy of a COH bond. And that just gives you some type of perspective on this. Okay? And of course, fatty acids are the building blocks of most lipids. Now, we talked about this the other day from this picture, and we talked about this with the structure of the cell membrane. The structure of fatty acids versus unsaturated fatty acids. The big component is the presence of a double membrane, or a double bond. Okay? Saturated fatty acids are called such not because they solidify at room temperature as commonly believed, but rather because they are straight. This allows them to stack together really nice and neatly. And it's kind of like paper, if you will, but by being nice and neat like this, they can stack really densely together. Whereas unsaturated fatty acids have the presence of a double bond. This covalent bond causes them to have a kink in them. And the presence of a kink causes them to fit loosely, just as you would compare crumbled paper. Obviously, straight paper will stack together nicely as opposed to crumbled paper. So we talked about this before, but that is saturated versus unsaturated fatty acids. Okay? And the difference has to do with the presence of a double bond. Likewise, we talked about the number of carbons that are present in these lipids as well. Lipids vary simply in the number of carbons that they have. And the longer the number of carbons, the more these chains can fold up and get knotted up, and also they tend to get thicker. Whereas the shorter chain fatty acids, they tend to be more volatile, which means they tend to evaporate a lot easier. In fact, we'll take a look at a chart right after this to see what we're talking about. This relates to petroleum. Of course, petroleum is predominantly composed of fatty acids, and mostly from ancient phytoplankton and zooplankton from millions of years ago. If you think about this, crude oil was made from these cell membranes of mostly the plankton from millions of years ago. Crude oil is these fatty acids compressed into a black tar substance when we pump it from the ground, and that is what we call crude oil. What we can do is distill it. That is, we use heat to filter out the different chemicals according to their density. We'll take a look at a chart, but if you recall in your chemistry class, when you heat things up, volatile substances float to the top, whereas dense, thick substances sink to the bottom. Let's look at this next chart here and look at this volatility. I'm not sure how well it shows up on your monitor, but this is the number of carbons. 1 carbon long in the fatty acid chain, all the way to 55 carbons in the fatty acid chain. This is a very long fatty acid chain here. Crude oil is composed of all of these fatty acid chains. When we heat crude oil up in a distillery column, we can separate all the different carbon-based compounds in there according to their density. The lighter stuff float to the top, whereas the heavier stuff sink to the bottom. Gasoline has a very loose definition, but it's typically any type of fatty acid chain that is about 4 to 12 carbons long. Diesel fuel is anywhere from about 7 to 24 carbons long, so it is a little bit thicker. But if you think about it, because this one has 24 carbons, diesel tends to have more energy density. You can actually store more energy in it. This is jet fuel here. We can move down to kerosene and heating oils. Fuel oils, you can see vary quite a bit. But look at the really heavy stuff at the bottom here. This is asphalt and pitch. Of course, asphalt and pitch are very thick substances, as are waxes. And the reason they are so thick is because they are so long in their chains. And the result is it causes those chains to knot up because they're so long. If we look back at these pictures here, imagine it being almost 50 carbons long. These would be so loose together, they would almost knot up. I mean, think about it. If you had long chains of string, you know those knot up really easily if you had them loose in your backpack or something. Whereas if they were shorter chains, you know they would have less of a chance of knotting up. That is why long chain fatty acids tend to knot up like that. I put this up as humor, but remember, when you fill your car up with petroleum, you are basically filling your car up with dead fatty acids of dead animals or even phytoplankton that lived millions of years ago. That's what crude oil is. Next, we want to look up the structure of a fatty acid, a cis versus a trans fatty acid. Cis fatty acid has to do with the way the hydrogens face. So this has to do with an unsaturated fatty acid. Cis fatty acids are those that have the hydrogen bonds on the same side. Okay? These are the type of fatty acids that are unsaturated that occur commonly in nature, and they tend to be healthy. You may again have heard of omega-3, omega-6 fatty acids. It all has to do with the presence of double bond, and it all has to do, of course, with how far that carbon is from the omega group. That's why we call it such. Take a look at this one here. You can see in this picture we've got a double bonded carbon, and you can of course see that there's two hydrogens on the same side. Because the hydrogens are on the same side, we call this the cis unsaturated fatty acid. By the way, I shouldn't have stuck the word poly up at the top because poly refers to multiple unsaturated fatty acids. Some fatty acids have two double bonds, like one here and one here, and that's what we would call poly. Because this only has one double bond, we would call this a monounsaturated fatty acid. That's pretty much it. Monounsaturated fatty acid. A polyunsaturated fatty acid has two double bonds built throughout it, and so this picture does not illustrate that. My apologies. Okay. Trans fatty acids, on the other hand, are unsaturated fatty acids that have the hydrogen on the opposite side like this. And again, we call it trans because the hydrogen is on the opposing side of the double bond. Now, that might not seem like a big deal. So what? Who cares if the hydrogens are on the opposing side as opposed to this side? But the answer actually has to do with what your body is able to recognize and digest. But it also does something else to it too. And if I go back to the cis fatty acid, do you notice the cis double bond here causes the kink to be induced? That's why there is a kink. It has to do with the double bond and the two hydrogens pushing it downward like this. But a trans fatty acid actually does not have a kink in it. And that is unusual. And not only does it not have a kink, the structure of the hydrogens to the side is not naturally occurring. Trans fatty acids are typically synthesized and they're synthetic. The problem is they're not easily recognized by the body, and ultimately trans fats do not digest or metabolize within our cells very readily. The net consequence of this is that these fatty acids tend to store up in our body and they don't break down very easily. They were made up long ago. In fact, I grew up when I was a kid where these fatty acids were actually used and touted as being a healthier option. But the simple reality is these fatty acids are much more likely to be unhealthy because they store up in your body and you don't have an enzyme that easily recognizes these fatty acids to break them down. So that is something to consider here with these. Trans fats are synthetic and your body does not have an enzyme that recognizes what to do with it. And therefore, even though it's supposed to be less calories for your body because you're not able to really break it down, the problem is these tend to just store up in our body. Fatty acids, for the most part, have been rid from a lot of our food products because they showed the acceleration of heart disease because of these buildups in our body. And unfortunately, I probably had a lot of this when I was growing up. I know movie theater popcorn was one huge culprit of trans fatty acids many years ago when I was a kid. Anyways, you can see the cis versus the trans fatty acid. It's simply whether or not the hydrogen is on the same or the opposing side. These are called monounsaturated fatty acids because there's one double bond, but if it had two double bonds within it, we would call it a polyunsaturated fatty acid. Hydrogenation is a process where we try to preserve food, and it's simply by adding hydrogens to these cis fatty acids and try to straighten them out. And the goal here is if we can take these unsaturated fatty acids and we can straighten them out, we can actually give them a different flavor. And we can also change the preservation of them because a lot of foods will spoil because of the unsaturated fatty acids, which are more of a liquid in their texture. And by the goal of this, of course, hydrogenation is, again, preservation and also the change of a flavor. And what we're trying to do here is, well, I'll show you. What we're trying to do here is take something that is unsaturated and we're trying to basically get rid of any of those double bonds and create these saturated fatty acids. So this is a process where we're essentially taking an unsaturated fatty acid and making it saturated. And by doing this, we are changing the preservation and the flavor as well as the texture of the food product. This is an industry process called hydrogenation. You might have heard of this process on some of your foods where it says no hydrogenated oils or something. That's what they're referring to, is the addition of hydrogen. Okay. And of course, if there's any double bonds left in here, they'll call it partially hydrogenated. It doesn't have to be the whole thing. All right, so those are fatty acids. We talked about saturated, unsaturated fatty acids, whether there's presence of a double bond or not. And we talked about if it has two double bonds or more, it's called polyunsaturated. And if it has the hydrogens on the same side, it's called cis. And if it's on the opposite side, we call it trans fatty acid. What if we wanted to chain these together and make a polymer? After all, you remember monosaccharides and sugars can chain together to form polysaccharides. Well, how do we take fatty acids and chain them together? Well, we can't chain them on end to each other. Instead, we can chain them into what's called a triglyceride. And if you look here, we have a fatty acid, but we actually have three fatty acids, all chained to a glycerol molecule at the very top here, okay? Triglycerides are also known as body fat. I probably should have wrote that in here, body fat. That stuff that's in your belly and your arms and all of that, okay? Triglycerides, body fat, is composed of one glycerol, which is basically 3 carbons, and it has 3 fatty acids connected to it. Like we said earlier, a glycosidic bond is what joins carbohydrates together, but an ester bond is what joins fatty acids to the glycerol together. Okay? This is a triglyceride. So you can see we've got 3 fatty acid chains, just like this. And here is a glycerol molecule at the very top. So here's the glycerol, and you can see how this omega end is forming an ester bond with those bonds there. And it's doing it, if you notice, in the exact same manner that the glycosidic bond formed. Where if you remember, there was an OH and an OH on both sides, and we got rid of the OH here, and we got rid of the hydrogen here. That left an oxygen linking them all together like this. This is how triglycerides form. So a glycerol is just a 3-carbon molecule. In fact, glycerol is actually a type of carbohydrate. But we can take this type of carbohydrate here, glycerol, and with 3 fatty acids of any length, we can make a body fat. Okay? So here's the formation of an ester bond. It is just like a glycosidic bond. You're taking the OH here off of the fatty acid, and you're taking just one of the H's off because, again, we are using the OH on the glycerol. That leaves behind the oxygen. This is just like what we saw with the glycosidic bond here. We call this an ester linkage, and that's what holds fats to glycerol, okay? And with that said, again, there's always just 3 of them because there's only 3 carbons in the glycerol molecule. That's what we call body fat, okay? We're going to go on to phospholipids, and that'll be one of the last things we do before we talk about steroids. But take a look again at this, and you'll see we have a possibility of attaching 3 fatty acids. What if we don't add a fatty acid here, but instead we put something else on this other carbon? That's what a phospholipid is. Take a look at this here. Phospholipids, they are what make up our cell membranes. And of course, they are the same thing as a triglyceride, but instead they have a phosphate instead of another fatty acid. So instead of 3 fatty acids on a glycerol, they have 2 fatty acids joined with an ester bond, and on the 3rd carbon they have a phosphate attached to them. You can see here's what's called a choline group. You do not need to know this though. Just know a phosphate is on the one end, and you can see it has 2 fatty acids for the other. So this is a phospholipid, and this is what makes up our cell membrane. Because we have the hydrophobic or nonpolar fatty acid tails, and we have a polarized hydrophilic phosphate, phosphates are really charged. That means we've got a charged side over here and a non-charged side over here. That means phospholipids are amphipathic. They're both charged over here and not charged here. And as you recall, that is essential to the structure of a cell membrane. As a result, these are amphipathic. The heads like water because phosphate is charged, but the tails do not like water because they are not charged. And again, can you sketch a triglyceride and a phospholipid? They both use glycerol, which has 3 carbons, but a triglyceride has 3 fatty acids, whereas a phospholipid has 2 fatty acids and a phosphate on that 3rd carbon. Okay? Triglyceride or fatty acid. Take a look. Can you tell the triglyceride from the fatty acid? Okay. Well, obviously, triglyceride has 3 fatty acids in it, whereas a phospholipid has 2 fatty acids, and on the 3rd carbon, it has a phosphate group attached. You can see that highly negative charge there on the phosphate, and the choline group is charged too. That's actually going to cause this to be polarized and hydrophilic, as opposed to the hydrophobic fatty side over here. Okay? The last thing we're going to look at is waxes and steroids. Now, we've been talking about these chains here. Waxes are a little bit different. Waxes are a lipid, which are nonpolar like all lipids. However, waxes are generally kind of plastic-like at room temperature. And you know this because you've probably dealt with waxes before. They have a high melting point, but they are malleable. And for the most part, we can use waxes as barriers in plants, and they're made of many diverse arrangements of fatty acids. The biggest thing you can notice up here with wax is it does not have an omega end to it. It's just a hydrocarbon. There's another one down here. It's still a hydrocarbon because literally all it is just hydrogens and carbons. You may have an oxygen group off to a side somewhere like this, but this is because it's just an omega end or something like that. Okay? You do not need to know all about waxes. Just be familiar. They are a kind of a barrier, and they can be used to like protect plants or your skins, or well, not on our skins, but like plants or your ears, I should say. And just know they are a type of lipid. course, these are things that we use wax as an industry. They are a lipid, and that means they do contain a lot of energy. That's why candles burn for so darn long. They have a lot of energy in them. Steroids, on the other hand, are the last thing we're going to look at. Steroids are confusing because when you look at them, they do not appear to be anything that we just talked about. I mean, when you look at a steroid, you're thinking, how does that look anything like what we just talked about? Well, it is composed of a hydrocarbon for the most part. Except you'll notice it has an OH off to the side here, and it has an oxygen off to the side. That red atom over here is an oxygen. But aside from the ends, the entire interior is hydrocarbons. Steroids are not proteins. They're hormones, but they're not proteins. Because steroids are fat soluble or lipid soluble, because they are a lipid, they can easily move with other nonpolar lipids. And that means steroids can easily go in and out of cells, and that makes them a very good type of protein messenger. Steroids generally activate protein synthesis on your DNA, which means these hormones go to your nucleus and directly tell the DNA what it wants to do. As we'll see with other hormones later, like insulin or growth hormone, those hormones are not steroids. They can't even go into the nucleus at all. They have to use secondary messengers to tell the DNA what to do. You can see generally it's made of 4 carbon rings, and it looks like a carbohydrate, but it's not. Again, why is this not a carbohydrate? It looks like a hexagonal ring, but that's because it does not contain the OH groups, the hydroxyls off to the side. It is at its core a hydrocarbon, and that makes it nonpolar. Okay, doesn't have any charges. So examples are cholesterol, testosterone, estrogen, progesterone, which regulates a woman's cycle, fertility and that. Steroids, again, have a variety of uses for them, and they're used all the time in our body. We do need these. And yes, cholesterol, you do need it as well here. Okay? To finish this up, we talked about carbs. Let's look at lipids again here. Are lipids generally polar or nonpolar? The answer is they are nonpolar. They are hydrophobic. They're not charged, and that's why they don't interact with water. What are hydrocarbons? And that is a compound that is composed of hydrogen and carbon. That's what lipids are. That's why they're nonpolar. They don't have any OHs. OHs tend to have charges. The structure of a fatty acid, if it's saturated, it is straight. Unsaturated tends to be kinked, but technically the definition is saturateds do not have double bonds in the hydrocarbons. Unsaturated have double bonds in the hydrocarbon, and in cis fatty acids that induces a kink. What do we call the bonds in lipids? It's called an ester bond. Compare a cis to a trans fatty acid. Cis has the hydrogen on the same side, trans are on the opposite side. What is another name for fats? Well, I meant body fats technically, because we have to be careful. Fat and lipids are not the same thing. But fats themselves would be a triglyceride. And if you sketch a diagram, you need a 3-carbon glycerol. And if it has 3 fatty acids, it is a triglyceride. If it has 2 fatty acids and a phosphate, it is a phospholipid. What are the use of waxes in organisms? Simply put, those are used to create a barrier. Plants use waxes. You and I don't really use them except in our ear, but waxes we typically use oils on our skin to keep us from drying out. A steroid is typically a 4-carbon ring, but you do not need to be able to draw this perfectly. I just want you to be able to remember this from your recollection. An example of steroids would be like cholesterol or testosterone, estrogen, progesterone. There's actually others out there like aldosterone. Off the top of my head, I'm sure there's several others. Just be aware of that a steroid is a type of hormone, but it is a lipid hormone, and as such, it behaves with the properties of a lipid. Okay? We're not going to do the foldable assignment, but I do hope this helped you out. I know this is a lot to cover here, but I hope this will be of some assistance to you. Have a wonderful day, everybody.